Yes. All right, fine. So yeah, so I was talking to you about this All India mock test. So this All India mock test is planned for tomorrow. Yes, everybody, am I live? Are you all here? Okay, am I here? I think that I'm live now. Alright, so this session it is meant to discuss and revise our applied biology in detail. Yes, and I was happy to tell you about this All India mock test. This is meant to happen, okay, to happen on 23rd Jan, that is tomorrow. So the event will begin by 10 a.m. So the advantage is that after you give the test, yes, after you yes Sonali, good evening. Yes, so after you give the test, you can have a detailed analysis of the question. So this time you will find it interesting. You could have seen in other tests also. We are going to provide you the video solution question wise. Okay. So if at all you have attempted, like you have a doubt with question number 71. Okay. You need not wait for the each and every question. Directly you can go and click the 71st question. You can view what is the answer. Also the detailed explanation in video. So even everyone we have given that complete video solution for our respective subjects. So that will really help you to boost up your preparation. Yes Ankita, yes Anishka, okay, yes Sonali. Come on everybody. So this is all clear to you? Fantastic. So yeah, so quickly we will revise the aspects of applied biology. So this is about your alcohol production. Okay, so alcohol is aerobic or anaerobic? Obviously alcohol is going to come under the anaerobic fermentation. It is not aerobic, but it is going to be, yes Amit, yes Anantara. Okay, it is going to be the anaerobic fermentation, not aerobic, it is anaerobic. Mainly saccharomyces helps in production of alcohol. Okay, so if you take the pathway, the glucose, it is going to get converted into pyruvate, and then from pyruvate, yes everybody, yes Rabita, absolutely. So then it is going to make your acetaldehyde. Okay, so finally ethanol is going to get produced. Got it? Okay, so let us do a question on this. So during the production of alcohol by means of fermentation using budding yeast, the oxygen supply, it is limited. Okay, why? Is it a fact that your budding yeast, they are obligate anaerobes, so they cannot tolerate oxygen, or they lose the mitochondria in absence of oxygen, or budding yeast, they are facultative anaerobes, or alcohol is going to get oxidized further in the presence of oxygen. So which could be the correct answer? Believe me, 4-mark question from this applied biology topic. Yes. Come on everybody. Yeah. Why your alcohol production, during the alcohol production your O2 level is kept low? What is their nature? Are they obligate or are they facultative? Facultative means little bit you can tolerate. Obligate means strict. See here, they are facultative anaerobe. Okay, so facultative means yes, they can actually grow up in the presence of oxygen. Consider your yeast. Okay, yeah. So let us just see which and all is coming under your obligate anaerobe. Obligate aerobes. Okay, right. Yes. So obligate aerobes, obligate anaerobes, right? Okay. So our yeast and many bacteria, they come under the facultative anaerobe. Facultative means it is either or choice. Okay, so either they will use the fermentation or they will use the aerobic respiration. Okay, so that is why you don't call them to be a strict anaerobe. Strict anaerobe means even if there is going to be little condition, that's all, they cannot actually survive. Yes, so got the difference. These are facultative, yeast as well as these bacteria. Yeah, so they are going to be the facultative anaerobes. Got it? So shall we just very quickly see through the examples of these? Okay. So if it is going to be Mycobacterium, they are the obligate aerobes. Okay. They are the strict aerobes. Who is obligate anaerobe? It is Clostridia. Yes. So these are coming under the obligate aerobes. Facultative means majority of them. Okay, so the yeast and other bacteria, they come under the facultative aerobes. Got it, everyone? I hope that this is all clear. Yes? Okay. So now let us have a quick look. Okay, so into this plant biotechnology aspects. Okay. Yes, everyone. Shall we just make up a move towards the next one? I hope that you are all ready to make it out. Fantastic. Come on, guys. So under the plant biotechnology, so we do have a number of important methods. So yeah, so we can generate so many transgenics. So very quickly we will recap these points. Okay, right. So the number of methods. The first method, it is going to be your Agrobacterium. Several questions are being shooted up every year on this Agrobacterium mediated gene transfer. Yes, it is all about your TI plasmids. Yeah, so the TI plasmids are seriously very important. These TI plasmids, they contain a specific region. So they are called as the tDNA region. Okay, so what your tDNA is going to contain? Okay, right. So tDNA, will it contain this opine catabolic genes? Do they contain opine catabolic genes? True or false? Do they contain opine catabolic genes? My tDNA, will it contain opine catabolic genes? Yes? No, they do not contain opine catabolic genes. Yes, Vidya. So what you can do is that you have to click the 3 dots that you see on the top of your YouTube session. Okay, there you have to go to this quality, go to this higher picture quality. Okay, so if at all it is asking in DP or this picture quality, you can increase the pixel. Okay, greater than 720 will give you a better view. Okay, yes, Vidya? Okay, fantastic. Right. Yes, Sangeeta. So, as far as this opine catabolism is considered, okay, catabolism means it is going to degrade. Okay, so this fellow will not help in the degradation. My tDNA will contain only the biosynthesis genes. What are they? NOS. Okay, oct. This is octopine. This is nopaline. So all these nopaline synthase, octopine synthase. So all sort of biosynthesis genes, it is only present within the tDNA. So my catabolism genes, it is present within the TI plasmid, but it is not a component of my tDNA. Got the difference? Okay. Then what is the component of my tDNA? My tDNA, it is going to contain the left border. Okay. What else? It is going to contain my oncogenes. Oncogenes, which are the oncogenes here? That is only causing the tumor, no? Yes, it is called as tumor inducing. TI is tumor. So tumor means oncogene. Who is the oncogene? Auxin and cytokinin. Okay. Apart from this, it contains the opine biosynthesis genes like my octopine, like my nopalin. Right. Then it has my right border. So can you see something is lying between this right border and opine? What can be that? Very good, Praveen. Yes, Sonali. What is lying between this opine and right border? I think 2020 question we saw today. It is a marker. Got it? Yes, usually markers are placed near the right border. So they will be get incorporated. Understood this? Yes, everybody? So this part is now clear? Okay, good. So where is going to be the virulence region? Virulence region, vir gene. It will all be present in the TI plasmid, but this is not within my tDNA. Okay, so this is going to be origin of replication. So what is in the tDNA? Only these factors. So what will be in my TI plasmid? So my vir genes, my origin of replication, my opine, catabolic things. So all these things are present. Yes, Anushka. Yeah, absolutely. Even I was. Don't worry, we'll compensate one more day. Okay. So these opines, they were of prime importance because they are going to be the amino acid derivatives. Okay. So this itself is going to serve as a nutrient. Okay. Right. And this agropine is also an opine. It is a sugar derivative. Okay, so these are all used by your atomifacients as a sole carbon and energy source. Yes, at my end it was a network issue, Anushka. So like no, that is why we were not able to yes, connect it. Absolutely. Yeah, that's okay. Don't worry, we'll compensate soon. Alright. So these opines, they are fantastic source of energy. Isn't it? So if you take it, this opines, they are going to be your amino acid derivatives. They are going to be the sugar derivatives, right? So they all help them to utilize them as the, oh my God, thank you Sangeeta, thank you Anushka. Right. So it is all going to utilize. Okay. Yes. Okay. So this TI plasmids, they contain the enhancer. Okay. Right. This enhancer is also associated with your right border. That is why they always say that this right border should never be deleted. Okay. So if the right border is deleted, that's all. Too many things will be lost. Enhancer will be lost. Your markers will be lost. Because all of them, they are associated only with the right border sequences. Yes. So this OCS, NOS, all these are the synthesis genes. So these are all present within your tDNA. Okay, so one time this question was asked, which gene is responsible for production of what? NOS, nopalin, OCS, octopine, okay, TRN, tryptophan, okay, right, and then FRS, fructopine, MAS, manopine, AGS, agropine. So these are all the variety of the things that is getting produced. Alright, okay, what is this? Mark this slide seriously, it is too much important. Many questions have already come up in the exam. In future also many will come. Okay, first what happens, you have to understand that the event is taking place in two places. What is that purple color one, pink color one? What is this? This is going to be the bacteria, or it is going to be your Agrobacterium. Okay, right. This other part, this is going to be the plant cell. Okay. Will this acetosyringone? Okay. So who secretes this acetosyringone? Who secretes the acetosyringone? Acetosyringone is an attractant molecule. Okay. So who is secreting it? Is it going to be your plant or bacteria which is secreting the acetosyringone? Acetosyringin is secreted by the plant whenever the plant is wounded. Okay. So as a response, yes Praveen, it is going to be secreted by the plant cell. Okay. So the plant cell is wounded plant cell, not normal. The wounded plant cell. Yes Rohan. The wounded plant cell is going to secrete the acetosyringin. So somebody should catch hold. Somebody should respond with this acetosyringin, no? So who is coming and responding? Who is catching hold of the signal? It is the Vir-A. Okay. So this virulent A protein or this virulent A gene, it is only going to have a receptor. This receptor is only holding the acetosyringin. Got it? Yes, this acetosyringin is held by this Vir-A. Understood? Fantastic. So now this is made a transport into this TI plasmid. Absolutely Anantana. So now it is moving to the TI plasmid. In TI plasmid, apart from the tDNA, you have the Vir region. This Vir region is going to get activated. Okay, and then it is going to produce Vir D1 and D2. Okay, so what it is producing? Vir D1 and D2. Right. So now what happens? This tDNA, it is now cut off. Where it will cut? Right border, left border. Okay, this is the region where your tDNA is cut. Okay. Once my tDNA is cut, yes, this Vir region, it is going to cap the tDNA. It should be protected. No, always you need a cap, isn't it? It should be protected. So this single stranded DNA, it is going to get protected. Now, all these F, D5, E2 and all, it is helping to make our tDNA to be safely transported from this bacteria to the plant cell. Got it? Yes, Samiksha. Fantastic. So from my bacteria, the DNA is slowly transferred into my plant cell. Okay. So now can we say what are the proteins that is involved in this bacteria? Which Vir? It is Vir-A, Vir D1 and Vir D2. Yes, Vir D1, Vir D2, Vir-A. F is also there, E is also there, B is also there. See here, B is only making the transport. Okay, so FEB and then A, DD. So you can remember already, you know this DEAB clue, isn't it? Okay, DEABF, DEABF. Okay, so this is all associated with the bacteria. So in the plant, which ones are participating? Who is coming inside? E2 is coming inside. Yes, D2 is coming inside. Yes, this is E3. So, only D and E, they are alone coming inside. Yes, F also. Yeah. So, all your B is standing out. So, inside the plant, who are there? D, E and F. That's all. Because this is all helpful in localization and all. No. So, these are alone helpful. Okay. So, now the total story of your Agrobacterium is clear. Yes. Many questions do come from this Agrobacterium. So, we will just have a look at few of the questions. Again, we will come back. Shall we? Very quickly, we will do some questions. Alright. Come on, everybody. All these are really important for your exams. We will do few questions. Right. So, the Bt toxin from this Bacillus thuringiensis is coming from which gene? Cry, Crow, CDC, Cree. Come on everyone. Bt toxin. Fantastic. It is the Cry gene. Okay. Right. What is Cree? Cree LOX P. Cree, it is a recombinase. Okay, CDC, cell division protein, you could have studied this in the cell cycle. Correct? Crow is not related here. Only the cry is the correct one. Alright. So, yeah, fantastic. So, right, this Agrobacterium, okay, TI plasmid vectors, they are used to generate transgenic plants. The following are the examples of the Vir gene encoded proteins that are too much important to transfer the tDNA. Okay, right. So this Vir-E, it is a single standard DNA binding protein. Vir-E. Think. Vir-D2 generates T strands. Vir-A senses plant phenolic components. Vir-F, it directs the T complex proteins for destruction in the proteosomes. Just now we saw a detailed look into the Vir as well as the function. Yeah, Sonali? Which is correct they are asking. Yeah, Rohan? Come on, come on. Where did we study this Vir-A? Vir-A is only binding with your acetosyringone. Acetosyringone is the plant phenolic. Definitely 3 is right. So D is eliminated. Am I correct? Absolutely. Right. And this one, but this one it happens inside your bacteria. Isn't it? Does it happen inside your plant cell? Fantastic. It doesn't happen inside the plant cell. Vir-A, I told you that it was inside the bacteria. We saw the diagram now, isn't it? Absolutely. So that is why, very good everybody. Awesome. Yes, Sonali, Rohan, Praveen. Fantastic. So they take place inside the bacteria. It is not inside the plant cell. So immediately you can eliminate the option with 3. All the others are gone. Only 1 is right. Yes. So yes, Samishka is going to be crygenes. So only 1 is right. That is your option D. Fantastic. Yeah. So Vir-E, it is going to make your DNA binding. Vir-D2 generates T strands. It cuts endonuclease. Yes, Anishka. Very, very happy. Too good. So D, it is going to direct your destruction. All the things are right except for your B. This is the incorrect option. Alright. Yes, everyone. Fantastic. Okay. See the next one. Agrobacterium tumefaciens is a gram-negative soil bacterium. Yes. True or false? Agrobacterium is a gram-negative soil bacterium. Okay, absolutely correct. Opine catabolism genes are present in tDNA. This point I described you in detail. Do you remember? See, this is a TI plasmid. Inside that you have your tDNA. Okay, right. So there is no poll here. Okay, think and answer. Opine catabolism genes, they are present in tDNA. No. What is present? Only this opine biosynthesis genes. Okay. So like your OCT, NOS, okay, opaline, all these biosynthesis genes are only present. Okay. Absolutely. So these are all catabolism genes. They are never present. So immediately you can eliminate the option which is going to contain the B. Isn't it? Yeah, absolutely great everybody. So the correct answer becomes D, goes without saying. Very good, fantastic. Yes, Rohan, Praveen, Anita, Anantara. Yes, Anishka, fantastic. Opines, it is synthesized by condensation of amino acids and keto acids. Absolutely correct. And a callus, it is formed by a crown gall without adding phytohormones. Why? You don't need a phytohormone. Already it is there in my tDNA, so it doesn't need any phytohormone. Got it? Yes, everyone? It is all clear? Okay, one more question we will do. See, applied biology is usually neglected, but seriously I will tell you guys, every year there is questions from applied biology, that too from the hot topic like this Agrobacterium. Okay, so never miss this for your examination. So following are the certain statements regarding the use of Agrobacterium in the plant transformation. Agrobacterium tumefaciens causes the crown gall disease, and this rhizogenous, it is causing a hairy root. True or false? Tumefaciens, they cause the crown gall. Rhizogenes, they cause the hairy root. Absolutely, it is a correct statement. Alright. Region A in Ti plasmid, it is responsible for replication, or the region D in the plasmid is responsible for virulence. Region means it is the vir thing they are telling. Vir A, is it for replication? Do you think that vir A is for replication? Never know. Vir A, it is for attachment with your acetosyringone. Okay, so this is wrong. Absolutely. Yeah, Shivani. Yes, Donna, Anushka. Yeah, Shubha. So this is absolutely absurd, no? So this can be eliminated. D is meant for virulence. It caps, isn't it? It caps your DNA like this. Absolutely correct. Oncogenic region, it is responsible for unusual amino acid synthesis. No, oncogenic region is meant for your auxin and cytokinin production. Yes, Anushka, C is the answer. So this is no, unusual amino acid is going to be the opine synthesis. Now you got the point, everybody? Yes, all the questions are seriously very, very important. Yes, so shall we make up a move? I think even in 2020 you got questions from this. So everything should be very, very careful. Right, coming to the next question. While designing an experiment for Agrobacterium mediated plant transformation, a student noted down the following points. So your part C, it is very well taken care of by applied biology. It will never let you down. Okay, so please go up with your preparation with this applied biology. Okay, so Ti plasmids and Ri plasmids, they induce crown gall and hairy root respectively. Correct or not? Yes, absolutely correct. So the options which do not have one must be incorrect. Immediately you can eliminate it. Yeah, Shubham, fantastic. Yes, Anantara, Suhani, Priyanka. Yes, Donna, fantastic. Give it up, everybody. Yes, Praveen. Yes, Anantara, fantastic. Okay, the enzymes octopine synthase, nopalin synthase, it is involved in the synthesis of octopine, nopalin respectively. This is a KG question, no? This option is right. Very good, fantastic. So you know that 2 is also right. This fellow can be eliminated. All the 6 genes, it is required for virulence. What is wrong with this option? Why this option is wrong? Have you ever studied C? No. A, it is helpful for binding your acetosyringone. B, it is helping in the transport from the bacteria to the plant cell. That is the role of B, isn't it? D, it is going to protect your DNA. That is D. Yes, absolutely. E, it is also helpful. F is also helpful for integration. Okay, E is helping in your transport and F is signaling and integration. That is F. G is also important. We could have seen G also. But C, there is no such vir C. Absolutely. Vir is important, Donna. See here, without vir, what will happen is that it cannot get the infection. Got it? This vir C is not wanted. Absolutely. Yeah, I got your point, Donna. Good. So this vir C is not at all required. Absolutely correct. Virulence C is not at all required because you don't see any vir C there. Yes, very good. Yes, keep it up. So we don't see any vir C there. So vir C is not at all important. So that is the reason why it is eliminated. So 3 is going to be absolutely wrong. Very good, everyone. You are all very smart. Good. So Samiksha, yes, Shubham, Manushka, Donna, Sangeeta, Praveen. Fantastic, fantastic. So this third option is eliminated. So obviously you come to know that D is the answer. So such questions, they usually help to solve very fast. Okay. By elimination you can solve fast. Okay. So the almost perfect 25 base pair direct repeat sequences, that is flanking your Ti and Ri plasmids, it is essential for tDNA transfer. Absolutely, it is a correct statement. Hence, D is going to be the correct one. See how many questions have come in the past on this. Yes, absolutely. Yes, everybody, come on. Yes. So many questions are there from this Agrobacterium. Alright, so shall we make a move towards the next question, everyone? Yes, everybody is ready. Don't worry, Anushka, you will do justice for part B2. Yes, maximize unit, maximum benefit. Yes, so this Agrobacterium tumefaciens, it is also known as natural genetic engineer. It causes the crown gall disease in plants. However, when the same bacteria, it is used to raise the transgenic plants with improved agronomic traits, no such disease was observed. Okay. Why? Yes, come on. Good. So why? It is because, yes, Vir D2, it is mutated in the Ti plasmid. Fantastic. Keep it up. Yes, everybody. Yes, Shubham, Donna, Praveen. Fantastic. So D2, it is mutated in the Ti, or the disarmed Ti is generally used. Heat shock during the transformation, it destroys the virulence. I told you, if virulence is gone, no infection. Okay, right, keep it in mind. Oncogenes are removed. Yes. Okay. Let us read this question one more time. Okay, I just got it. Okay. So, yes, the Agrobacterium, whenever it is used to raise, no tumor is produced. Why there is no tumor? Because this is for sure. You have removed the oncogenes. In that place only your new genes are sitting. Okay, so definitely D should be there in your option. So these two are eliminated. Okay, so Vir D2 gene is mutated. Do you think so? Or the disarmed plasmid is used. What do you mean by disarmed? Disarmed means you are going to remove something. What you will remove? You are removing the oncogene. That is what is called as disarm. Vir D2, it is helpful to carry your DNA. If it is mutated, will it carry? No, absolutely. So that is why this option is eliminated. Which one? Your D2 is mutated. You cannot make. Yes, absolutely, Shubham. It is disarmed. Same meaning only. Disarm is going to be the removal of the oncogene. That is what is called as disarmed. But D2 is very much essential for your transit. Okay, so it cannot be mutated. So A is eliminated, and heat shock destroy virulence. I told you virulence is very important. You can never destroy them. So the correct answer becomes D. Fantastic, everyone. Don't worry if you are wrong. You can learn. You are ultimately here to learn. Okay. Which of the following is used as a selection marker for the cells that is transformed with the Agrobacterium? I'll read out. I think the options are a little bit dull. Neomycin. First one, it is going to be the neomycin phosphotransferase. Second option, it is streptomycin phosphotransferase. Third one, it is hygromycin phosphotransferase. Believe me or not, this question has come in many examinations. Apart from a CSIR, all over DBT, ICAR, so many, this has been very common question. GATE, plant BT, all this question is very common. Absolutely. So, your Agrobacterium is very good selected by the hygromycin. It is a specific, yes Anantara, yes Shubham, Sangeeta. Fantastic. It is going to be the hygromycin phosphotransferase. But along with that, what other things can also be used? Okay, this is very commonly used. Okay, but apart from this hygromycin, you can also use the neomycin. You can also use the streptomycin for the marker. Oh, yes. That's okay. Yeah. So, absolutely. So, see here, hygromycin is the maximal one, maximum used one. But apart from that, all the other things can also be used. If the question is, which is going to be the most used, definitely I should have gone with 3. Got it? Got the difference? But the question is, which are all used? Anything can be used, like within the given range. Neomycin, streptomycin, these are all can be used. Yes, Donna, correct. So the correct answer should be your answer D. Okay, so if the question is little bit modified, that which one is very efficient, which one is most commonly used, maximal used, then you have to go with hygromycin. Understood the minor thing? Okay. So Vir genes, it is required for the tDNA transfer and processing. Where it is located? It is located on the tDNA, outside the tDNA, on the plant genome. None of this. We can eliminate none of these. Yes. So where is the Vir genes present? Where is the Vir genes present? Fantastic. Yes. Okay. Very good. Shubham, Sonam, S. Kantal, Anantara, Rohan. Fantastic everyone. Yes. I told you in tDNA, maximum you have the oncogenes like your auxin, cytokinin. You have your octopine synthesis gene. You have the right and the left border. That's all you have. Okay. So outside it only you have the virulence gene. You have the opine catabolism gene as well as this origin gene. Okay. Yes. So absolutely very good everybody. Yes Anushka. Yes Shuhani. Sonali. Fantastic. It is outside the tDNA region. Oh yes. Seriously, time flies in the class, guys. Yes. It's almost time's up. Okay. So maybe the other questions we will make it. This one alone we will do very fast. Okay. So in response to the activation signal, an endonuclease, it is creating nick in the DNA. So who is creating the nick in the tDNA? Definitely, yes Anushka. Definitely C is eliminated because there is no such fellow called C. What is A? A is meant for binding with your acetylsyringone. B is meant for transport. So who is the endonuclease? Only D is the endonuclease. Very good. Yes Anushka, Kuntal, Shubham, Rohan, Anantara. Yes Anushka, fantastic. It is going to be the D. Yes, absolutely. It is only going to make a cut at the endonuclease. Okay. So this right segment for this octopine tDNA, it is absolutely too much necessary. Why it is necessary? The right segment. Yes Preeti. Yes Almas. Good. So the right segment is necessary for what? For the agropine biosynthesis, for the tumor formation, for conjugation, all of the above. Let us see. Okay. So what happens? Right. Let us come here. Okay. So I think we should go back to the first slide for this. Yes, just a minute. I'll go to the first slide. I'll show you the structure. Okay, fantastic. See, this is my right border. Who is near the right border? Who is near the right border? Can you see? This is opine. Got it? Okay, absolutely. So what happens? Your right border, it is going to have, okay, so have the counterparts from your opine biosynthesis. Okay, so this tumor formation, it is near your left border. Okay, so left border only auxin, cytokinins it is present. Okay, then only opine, then only it is going to be your right border. So near the right border, who is there? Opine. Yes, that's okay. So it is going to be the opine biosynthesis. Tumor is with the left border. Okay, right. So you don't want it, you will remove them also. So that is why left is always not considered to be so important. Right is only important for your, yes. So right is only important for this. Got it? Yes? Alright, fantastic. Okay everybody, so we have come to the end of this session. Yes? Okay, so all these things it is done, I guess. Okay, fantastic everyone. So have a good night, sleep well, and tomorrow, yes, there is the mock test. So give it with the Josh. Yes. Yeah, right border it also helps in the transfer. Correct. Right border helps in the transfer. But among the given option, yes, this is more suitable. Yes, I agree with you. The right is very important for your marker. It is very important for your transfer. It is very important for your opines also. Okay, but auxin and cytokine is not so important. So it cannot be all of the above. Okay, so if I have an option A, B, C and D, if it is only B and C, I cannot say it as all of the above. Got it? Yes? That is how this is going to be eliminated. Got it? Okay, fantastic. So this is AIMNet. Yeah, sweetie, helping you. My ID is biovsa16@gmail.com. Alright, fine everybody. Keep it up. So this is for AIMNet All India mock test. Okay, right. So this mock test is giving you a wonderful experience. So please do enroll at the earliest, and I really want you to win these amazing prizes, right? And the link will be opened by 10 a.m. After that you will get the fantastic analysis too. So it is based on individual question wise. So if at all you have a doubt at a particular question you might be knowing, you can directly go to that and have a clearance. Video solution is given. Alright? Okay, fantastic everyone. So all the very best, and definitely use my code Saranya Live if at all you are using it for the first time. Alright, thanks a lot everyone. Take care. Yes, see you soon tomorrow.